All right, hi everybody. Today we are going to be talking about macromolecules, and specifically, we're going to talk about the carbohydrates and the lipids. We will talk about the proteins and nucleic acids in the next set of notes. So make sure you have a paper and pencil ready to go. And please, if you start tuning out, take a break. Pause the video, go run outside for a few minutes, and come back inside before you move on. Don't miss out because these are important notes. All right, so let's talk about what macromolecules are specifically. Obviously, the word macro as opposed to micro means that they're big. So, proteins, for example, are the largest molecules we know of in the universe. They're made of a few very specific atoms, and we're going to talk about what those atoms are, and they are what accomplish all of life's functions. They're put together in a very special way, and we'll talk about that. And they can be incredibly complex, so complex that we often need computers in order to analyze them. This is an image of a protein. Each one of these little colored dots represents a different atom, and this is just one protein. And obviously, we've got DNA as well, and each one of the little colorful dots represents a different atom in DNA as well. You have 3 billion base pairs that make up your DNA. All right, so there are four main kinds of macromolecules that are part of all life on our planet: the carbs, the lipids, the proteins, and the nucleic acids. So, to build macromolecules, most of the macromolecules are made of little parts that are called monomers. So, imagine like a brick wall or a stone wall. You have a wall and it's made of all these different building blocks. Those are the monomers. They're the simplest unit, and you put a whole bunch of them together and you get a polymer. The prefix mono means one, the prefix poly means three or more. Lipids are the one exception to that rule. They do not have monomers. So lipids are just big molecules that don't have little building blocks that you put together to build them. The movement between monomers and polymers is facilitated by adding or removing water, and that's what we're going to talk about right now. So there's two processes. They are the exact opposite of each other, and they're called dehydration synthesis and hydrolysis. So dehydration, the name is really sensible. If you're dehydrated, it means you're low on water, and to synthesize something means to build it. So in this circumstance, look very carefully at the diagram. In this circumstance, I see an H on the left and an OH on the right, and they get removed to build that happy water molecule. And because they're removed, now the purple circle on the left, the purple atom on the left, and the purple atom on the right, they're missing something that they need to bond with, so they bond with each other. And so by removing water, by removing an H from one and an OH from the other, they join together and we have synthesized a larger molecule. So that's putting monomers together to make a polymer, and that's called dehydration synthesis. We are increasing the complexity. That's called an anabolic reaction, and it requires the input of energy, and that's an endergonic reaction as opposed to an exergonic reaction. And it requires enzymes. I'm going to tell you just a little bit about enzymes. We're going to learn all about enzymes in another unit, but let me just tell you a little bit about how enzymes work. Enzymes speed up these reactions. So they make reactions in your body happen a gajillion times faster. Enzymes are proteins, and they have very specific shapes. And they fit very exactly with a certain type of molecule. And if that molecule is somehow changed, then the enzyme doesn't fit it. Or if the enzyme is changed, let's say you have a mutation, and the enzyme is a funny shape, then it doesn't fit anymore and it doesn't do its job. So there are enzymes that put molecules together, that build complexity, and there are enzymes that pull molecules apart. And that's the opposite of what we're talking about here. So this is dehydration synthesis. The opposite process, called hydrolysis, can also happen. Look at the happy water molecule. It's being added into the larger molecule at the top, so that H and that OH are getting replaced, and now that molecule has broken apart. Imagine that you're digesting sucrose, you're digesting sugar into its component parts. It's being broken apart by the addition of water. And an enzyme helps that to happen. So in this case, we've reduced complexity. So that's called catabolic in that case. It releases energy. So imagine digesting food. Now you've got the energy from the food. So that's an exergonic reaction, and enzymes are required. Enzymes are required no matter which direction you're going. We're going to learn lots more about enzymes as we go. All right, so now we're going to take a tour. Things you want to focus on. You want to focus on the structure of these molecules. You will need to know their function. Are they storage molecules? Are they information storage or energy storage molecules? What do they do? What are their functions? You need to know their monomers. Only lipids don't have monomers, so monomers are the building blocks. What atoms are needed, and then what are their polymers, their bigger molecules. It is easy to get confused, so don't hesitate to ask questions, even though it's a video and you can't actually ask questions. You'll have to ask me questions later. All right, so let me back up. We're going to focus right now on carbohydrates. I love the name carbohydrate. It kind of gives you a sense of what is involved in these molecules. They are all made of carbon, hydrogen, and oxygen, and nothing else. So if you see a nitrogen, if you see a phosphorus, if you see a sulfur, it is not a carbohydrate. So carbohydrates very specifically are the sugars and the starches. They are made of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio in the monomers, and it's very close to a 1 to 2 to 1 ratio in the polymers. And that'll make sense in just a minute. So these guys are used for short-term energy storage. So imagine, it's not necessarily a true story, but sometimes parents will say, oh, I gave my kid candy before bed and they went crazy and they were running around the house. And so a lot of times parents think that sugars give kids lots and lots of energy. So short-term energy, that's what the sugars and starches are for. Long-term energy is fat, and that's lipids. And we'll talk about that in just a second. All right, so the monomers of all carbohydrates are called mono. There's that prefix that means one. Saccharides. So a monosaccharide. The word saccharide means sugar. So a monosaccharide is a single sugar. And you can put the monosaccharides together to make disaccharides, or you can put several of them together to make polysaccharides. So different sugar monomers can have different numbers of carbon. So we're going to, down along the bottom here are several different examples of monomers that are carbohydrates. And one thing that is kind of handy is that it is a good review of your functional groups. So take a look at these. Notice that these two sugars are all have 3 carbons, 6 hydrogens, and 3 oxygens. However, they are slightly different because they're carbonyl groups are located in different positions. So the carbonyl group is on one end. Remember that means it's an aldehyde if it's on one end, whereas the carbonyl group is in the middle, so that makes it a ketone. So these are just 2 examples. Notice they have the carbons, and then so 3 carbons and 3 oxygens and twice as many hydrogens. That's very, very characteristic of a monosaccharide, the monomer for all carbohydrates. So this is one example. Here's another example. Ribose is a super important sugar that's important for DNA and RNA. So this is a pentose. It has 5 carbons. So 1, 2, 3, 4, 5. And now let's look at how many oxygens. 1, 2, 3, 4, 5. Oh yeah. And I'm guessing that there are 10 and there are hydrogens. So there's that 1 to 2 to 1 ratio that we were talking about. This is also a pentose sugar. It's got 5 carbons, but this one has the carbonyl group in the middle, and so we call that a ketone sugar instead of an aldehyde sugar. And then this is another one. These are the most famous sugars when it comes to carbohydrates, the monosaccharides. So we've got glucose, galactose, and fructose. Notice that all 3 of them have the same molecular formula, C6H12O6, but where that carbonyl group is varies, and in this case, where the hydroxide group is varies. The other thing that I'm going to mention, and they'll say it in the notes in just a second, but most of the time when these sugars are written out, they're drawn in a ring shape. So for example, this would be a six-sided ring, and the carbons are all bonded to each other. But what they've done in this drawing is they've taken the ring and they've just let it go so that it makes a straight line, but it is capable of tying to itself, which we'll see in just a second. So usually you don't see glucose as a long chain of 6 carbons. Usually you see it in a ring structure. But I want you to know that it is possible to have it stretched out. So these are the major carbohydrates used for energy, the monosaccharides and the disaccharides, the hexose sugars are the most famous of the monosaccharides. So those are the ones we just talked about on the last slide, glucose, galactose, and fructose. Glucose is the sugar that is made during photosynthesis. So it is probably the planet's single most important sugar on the whole planet. Fructose is the sugar that makes fruit sweet, and why we like to eat fruit and why animals like to eat fruit, because of its sweetness. Okay, they are typically shown as carbon rings. So I was going to show you this diagram right here. So on the left is what we just saw on that previous slide, where they're all kind of lined up. The carbons are all lined up, but they're showing you here that, and notice the arrows going back and forth, it is possible for them to bond to themselves. So for this long chain to fold and to bond to itself and to form a ring. The molecular formula is still exactly the same. This hydrogen just moves from this oxygen over to this oxygen, but the molecular formula is exactly the same. So you get that hexagon shape in that case, pentagon, hexagon. This one's a hexagon shape. All right. The monomers combine by dehydration synthesis. So when you take 2 of these single sugars and put them together with dehydration synthesis, you remove a water molecule, you get a disaccharide. The prefix di means 2. So there are 3 really famous disaccharides. If you put 2 glucose molecules together, you can get maltose. So that's the sugar if you're having a malt. Glucose plus fructose is what makes sucrose. So that's what you make cookies out of, or Kool-Aid. That's table sugar. And then glucose plus galactose is where we get our milk sugar. So that's lactose. Did I say that right? Glucose plus galactose makes lactose. All right, and then I want you to study these diagrams here. They're so intimidating at first, but your brain will start to wrap around them as you look at them more. So here are the ring structures of 2 glucose molecules. So it's very repetitive. It's exactly the same shape 2 times. But on this one, we have highlighted an OH group, a hydroxide group, and on this side, we've highlighted just the hydrogen by itself. And we're going to remove the OH and the H to build ourselves a molecule of water. Well, now this carbon that's right here is missing a bond, and now this oxygen is missing a bond. So this oxygen is going to bond with this carbon, and so that's what you see right here. Here's the oxygen bonding now with this carbon. So we had 2 monosaccharides. We removed water through dehydration synthesis, and they glued together to make a disaccharide. The disaccharide in this case is called maltose. All it is 2 glucose molecules stuck together. Okay, and we can do exactly the same process, but notice that fructose is a pentose. Fructose is a pentagon, so it's a 5-sided shape, so it's got a slightly different shape. So I want you to think about this because it's super cool. You have enzymes, remember, that are putting these molecules together. And notice that these molecules are different shapes. So you need different enzymes to put them together, but you also need different enzymes to pull them apart. So if you're eating malt sugar, you need an enzyme that will break it apart into glucose and glucose, but you need a different enzyme to break the sucrose. Here's where it becomes interesting. How many of you are lactose intolerant? You can be lactose intolerant for several reasons, but some of the folks that are lactose intolerant, it's because their enzyme that breaks, now this is sucrose, this isn't lactose, but their enzyme that breaks up the lactose molecule into its 2 monosaccharides isn't functioning properly. It doesn't fit correctly, and so they can't digest that food, and it makes people feel crummy when they have the lactose sugar in their diet. All right, moving on. So we've just talked about the single sugars are the monosaccharides. Through dehydration synthesis, we can put two of them together to make disaccharides. And then through lots of dehydration synthesis, we can build our polysaccharides. Polysaccharides are very large molecules. They are made of many sugars. That's what the word polysaccharide means. And there's two main uses for the polysaccharides. They can be used to store energy. It's not as much storage as like a fat would be, but it's some storage of energy. Remember that plants are making glucose. I make it a ring. They're making glucose during photosynthesis. So they're capturing the sun's energy and putting it into a food molecule. And then plants and animals put these molecules all together to make this like big molecule that stores energy. So there's the energy storage ones. If it's a plant, it stores it as a molecule we call starch, but the scientific name is amylose. And if you look at amylose, look at this diagram carefully. Each of these little green things, that's a monosaccharide. That's glucose all stuck together. There is another storage molecule that's called amylopectin. And the only difference, notice it's still all these glucose molecules put together. The only difference is that it branches. So amylose is just one long chain of glucose molecules. Amylopectin is branched. Similarly, animals are capable of putting glucose molecules together during dehydration synthesis, but theirs is all branched. So they don't make this long chain of starch molecules. They make a branched chain, and that one is called glycogen. But it's still energy storage, and every single one of those are just glucose molecules, just a bunch of glucose molecules put together during dehydration synthesis. All right, and then the other reason or way in which these polysaccharides are used is for support. So cellulose is the major component of plant cell walls, and it's a whole bunch of glucose molecules put together. So plants, remember, don't have skeletons, so they need cell walls around their cells to allow them to stand upright. And those cell walls are polysaccharides called cellulose, that just a bunch of glucose molecules put together. Now, here's the part that I think is super cool. Oops, I'm going to back up a second. The difference between starch and cellulose has to do with the way the glucose molecules are linked. So look at my hands. Here we go. Let's do it this way. Can I do it this way? There we go. Well, look at my thumbs. We'll do it this way. So for starch, the glucose molecules are linked like this. Look at my thumbs. They're linked and linked and linked and linked like this. You got a whole bunch of, notice my thumbs are up in the air in this case. For cellulose, the glucose molecules are linked like this, one up and one down, and then one up and then one down, and one up and one down. So each glucose molecule, every other molecule is flipped upside down. They're still glucose molecules, but they're arranged in a slightly different pattern. Now, I want you to think about that. I've been telling you about shape and how important shape is to digestion, to putting molecules together, to pulling molecules apart. So let's see if you can start to figure this out. Here is what starch looks like. So notice if you look, these are all glucose molecules joined together. Look at the OH groups because that can kind of catch your attention. They're highlighted in yellow right now. And each one of those hydroxide groups is pointing down. Now look at where the hydroxide groups are in cellulose. These are still glucose molecules. All of them are glucose molecules, but they're flipped upside down from each other. So one is at the bottom, one's at the top, one's at the bottom, one's at the top. So now I want you to think about if you ate starch or if you ate cellulose. Well, starch is a sugar that we have, a polysaccharide that we have in all kinds of foods. And we have the enzyme that breaks this bond and allows us to pull the glucose molecules out. We digest it, we get great energy from our starches. But what if you were to eat cellulose? Well, cellulose is what's found in grass and in leaves. If you or your dog were to eat grass or leaves, you know from your dog what's going to happen. Dogs, when they eat grass, puke it back up. Why do they puke it back up? Because they do not have the right enzyme for digesting cellulose. Cellulose is full of tons and tons of energy. It should be just as good a source of energy as starch is, but we don't have the ability to digest it. So carnivores can't digest it. So herbivores have the same problem. different types of herbivores have figured this problem out. So how are herbivores able to eat grass and leaves? So there's several different ways. So first of all, we've got the termites. Termites have a symbiotic relationship with a protist. Now, a protist is a whole other kingdom of organisms. It's not an animal, it's not a bacteria, it's not a fungus, it's a whole other kingdom of organisms. And protists, certain protists, have the enzyme for digesting cellulose. So termites, they can't digest wood, but they eat the wood, and then the protists in their guts are able to digest they have the enzymes for breaking the cellulose into its component glucose molecules. And in the process, they feed themselves, but there's so much left over that the termites get fed also. So they're helping each other out. We call this mutualism. So the termite is providing a safe place for the protist to live, and it's feeding the protist. And then likewise, the protist is breaking these molecules up and feeding the termites. So they're helping each other out. All right, ruminants like cows, they have a special digestive tract that allows them, that helps them to digest this really complicated cellulose molecule. They don't have the enzyme either for digesting grass. So they, number one, have a super complicated upper GI tract that does contain a bacteria that does have the enzyme for digesting cellulose. But in addition, and this is gruesome, cows, they'll eat all kinds of grass, and then they'll go and they'll sit down or they'll just chill for a while. And while they're doing that, they puke back into their mouths. It's called chewing their cud, and they just kind of chew up their puke. And what they're doing is that they've got these really long cellulose molecules and they can't digest it. Their bacteria helps a little bit, but by chewing it, they're actually their teeth are pulling the glucose molecules off one at a time. Instead of having an enzyme that removes the glucose molecules, their teeth are cutting the glucose molecules apart. But it takes lots and lots and lots and lots of chewing and lots and lots and lots. It's a very inefficient form of digestion. And then there are the Sichophores, and we all think that bunny rabbits are cute and wonderful, and I used to have a bunny rabbit too. Sichophores, they don't have the enzyme, they don't have a bacteria to help them. Instead of having a really advanced upper GI system, they have an advanced lower GI system. So their bodies work really hard to digest cellulose, and then whatever is not successfully digested is pooped, and then they eat their poops. It's not really poop, to be fair. They're called cecal pellets, and cecal pellets, they get to re-eat. So as they poop them out, they'll re-eat the cecal pellets. And then once it's fully digested, then the pellets look like that, and then the rabbit won't eat them anymore. Isn't that pleasant? So I just want you to get the hang of it, though, that cellulose, the leaves and the grass, they're so full of energy. If humans had that enzyme to digest, we would never go hungry. I'm not saying that we want to eat grass or leaves, but in starvation situations, if you could digest leaves or grass, you would never have to worry about starvation. So it's just a crazy thing. All right, other important carbohydrates. So let me back up. Remember that carbohydrate, carbon, hydrogen, and oxygen, they include monosaccharides, disaccharides, and polysaccharides. So the polysaccharides we talked about, starch we can digest, cellulose we cannot digest. Then there are other important carbohydrates, polysaccharides, like chitin. Chitin makes up the cell walls in fungi and in insects and in dissolving stitches. That's not at all important. But so the skeleton of a bug is made of chitin. And then there's another important macromolecule called peptidoglycan, and that is a polysaccharide also. And that's what the cell walls of bacteria are made of. It's called peptidoglycan. All right, that is it for carbohydrates. Now we're going to talk about lipids. It might be a good time for you to pause the video and step away. Review your notes, perhaps. Take a moment to look at your notes and see if you can remember things that we just talked about. Pause, whatever you need to do, and now I'm going to go ahead and continue. All right, so let's talk lipids. First of all, lipids are fats. So they're the fats, the oils, and the waxes. They are also made of carbon, hydrogen, and oxygen, and so you should recognize them. However, it's not the 1:2:1 ratio that we saw in the carbohydrates. Carbohydrates have much more oxygen than the lipids do in general. These guys are used for long-term energy storage. So these are your fats. They're also used for insulation. So fat can be used to keep animals warm. There are no polymers. So remember that we had monosaccharides that could be attached to make disaccharides that could be attached to make polysaccharides. There's no such thing with the fats. They don't have little building blocks. And there are three major types that we're going to talk about today, and then we'll be done. There are the triglycerides, the phospholipids, and the steroids. All right, triglycerides are kind of a bad thing. I mean, they're good. We have to have them. Fat is not bad unless we have too much of it, and then it's bad. So we need some fat to be insulation. We need some fat to use when we're not eating so that we have a source of energy. So fat is not in and of itself bad. It's when we have too much of it that it's bad. So you might have tests, especially as an adult, you might have tests where they do lipid panels. And what they're trying to do is assess how many triglycerides are in your blood. And high levels of triglycerides are they go along with being obese, and they indicate a propensity for diabetes, heart disease, strokes, all kinds of problems. So we don't want to have high triglycerides. So the prefix tri means three, and then glyceride refers to one of the molecules. It's right here. And this molecule is always annoying to students because it's not one of the macromolecules, but you kind of need to know it. So it's called glycerol. It's got 3 carbons and then 3 hydroxide groups. And the hydroxide groups are super important because this is what a fat looks like. So it's a long chain of carbons and hydrogens. Notice the difference between it and a carbohydrate. A carbohydrate would have had lots of oxygens among these. There are almost no oxygens. The only oxygen is right here in the fat. So very, very, very fewer oxygens compared to all the carbons and hydrogens that we have here. So now watch what happens. This is a fatty acid. It can, through dehydration synthesis, attach to a molecule of glycerol, and actually 3 of them. There's another hydroxide group here, and there's another hydroxide group here. So you can actually attach 3 of these fatty acids to a molecule of glycerol. Those are called ester linkages. You don't necessarily need to know the word ester linkage, but it could come up in a test question. I'm not going to ask you what is an ester linkage, but it might be referenced in a test question, not asking you what it is, but just in ester linkages in blah, 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 blah. And you'll be like, what's an ester linkage? Well, that's what an ester linkage is. All right, so now let's talk about the fatty acids themselves. So this is a lipid, and it's got a whole bunch of carbons and hydrogens. And at the end, there is also, for the fatty acids, there's always a carboxyl group. That's what this is on one end. Before it joins with the glycerol molecule, there's always a carboxyl group. Now, there's the difference that you've probably all heard of, the saturated and unsaturated fats. So what we're talking about with saturated and unsaturated fats is just whether they are saturated with hydrogen or not. So the difference is whether they have single or double bonds. So an occasional double bond between the carbons prevents them from bonding with hydrogens. And so there are fewer hydrogens in an unsaturated fat. That'll make sense in just a second. Let's see if there's even an example. There's not a good example on this page, but listen up and I'll give you some ways to remember. So it says saturated versus unsaturated refers to the bonding of carbon in the fatty acids. Saturateds have no double bonds between their carbons. So take a look at this one right here. This is a saturated fat, and I can tell because there are no double bonds between the carbons. There is a double bond between a carbon and the oxygen in the carboxyl group. That doesn't count. There are no double bonds between carbons, so this one is a saturated fat. I wish they gave an example of an unsaturated fat here, and they didn't. Okay, so unsaturated have at least one double bond. So imagine one of these, let's say this carbon here has a double bond. That would mean these two hydrogens wouldn't be there because they're bonding with each other. They can't bond with those hydrogens. So it's unsaturated. It's not saturated with hydrogens anymore. So it influences the shape of the molecule if it has a double bond. Every time there's a double bond, it causes a kink in the fat. And so you can see that in this diagram, and we'll get to that diagram in just a second, but it causes it to bend. It also affects whether they are solid or liquid at room temperature. So this is how I help my students remember. Write this down, underline the S's. Saturated fats are solid at room temperature, and they have single bonds between the carbons. The other thing that I try, it's a weaker link here, but they make you sick. Not really, but they're less healthy, so they're not as good for you as the unsaturated fats. So unsaturated fats are everything else. Unsaturated fats are liquid at room temperature. They're healthier. They have a double bond. They don't have all single bonds. All right, so let's look at a close-up here. So saturated fat, I said, are solid at room temperature. So think of butter, or maybe some of you know Crisco. Unsaturated fats are liquid at room temperature, so think olive oil. They have all single bonds, so look at the single bonds here, as opposed to there's a double bond here, and the double bond causes the molecule to bend a little bit. So unsaturated fats have a different shape to them, and that's what causes them actually to be a liquid. All right, next type of lipid that we're going to talk about are the phospholipids. And as maybe you can guess, phospholipids have a new element that we haven't talked about yet. They have a phosphorus atom actually embedded in a phosphate molecule. So they're going to have a phosphate molecule attached to them. So it says here that they are modified triglycerides. See if you can see how they are called modified triglycerides. Here are the fats. We just saw those a minute ago. So there's two fats this time. There's two fatty acids attached to, here's the glycerol molecule right there. So there's two fats. But this time, the third, instead of having a third fat, it's attached to a phosphate molecule. Now, that's super important because a phosphate molecule, you can see with the minus right there, it's actually ionic. It's one of our common ions. Remember way back in chemistry class, phosphate was one of our common ions. So that means that there's a charge right here on this molecule. All of the other lipids are nonpolar. That means that water is not attracted to them. They don't dissolve in water. But phospholipids are partly polar, isn't quite the right word, but that's what we say. So we say the head is polar. It's technically ionic, but we'll go with polar. And the tail is nonpolar. So the head, we often draw the head. Let's see, I'll show it down here. Let's see if I can close up on it. We often show the head of a phospholipid, the part that is polar, we show it as a circle. And then we show the two fats coming down as these two lines. So it looks, whoops, excuse me, it looks a little bit like a stick figure that maybe a 2-year-old would draw. It's got a head and two legs, no body, no arms or anything like that. That's how we kind of cartoon a phospholipid. So let me go back where I was. Excuse me. All right, so the head is attracted to water, and the two fats, they are repulsed by water. So I'm going to show you how this works. So it's a 3-step image, and I want you to look at it. So let's see if we can scooch in here. Hold on. Oh, and I forgot one thing. It says that they are the major components of cell membranes. Phospholipids are what make up cell membranes, super important. And they are arranged in a bilayer. So that means there's 2 layers of them together. So here are the two fats. The fats face each other, and then the heads point away from each other. What? Right. So let me show you this diagram to see if I can make sense. So here's a cell, and this is really important that you get this. Outside of a cell, there's water. Maybe that water is blood. Maybe that water is something else. But there's liquid outside of a cell, and then there's liquid on the inside of the cell. So these molecules that are called phospholipids, the heads, which are represented by this red line, we'll look at it up close in just a second. But the heads are represented by this red line, and the tails are represented by the yellow. The heads move toward water. So there's a head near the outside of the water, the outside of the cell, and there are heads that are near the inside of the cell. But the tails move away from the water, so they go to the inside of the cell membrane. So let's break that down. Here's the cell membrane again. We're looking at it a little bit close up now. You still can't see the actual phospholipids yet, but you can see all kinds of other things that are involved in the cell membrane. We're going to learn more about those in the next unit. Let's see if you can see how that works. So this green box right here, this green box, we're going to focus in on that. We're going to look at that even closer. And now you can see the phospholipids. There are the red heads and the yellow tails. The red heads are pointing toward water, either the inside of the cell or the outside of the cell. And the tails have flipped in. It sounds dumb because you can't really do this, but I'm trying to make a point. If I had 100, more than that, but whatever, 100,000 phospholipids in my hand, and I just had a pile of them, and I threw them into a bucket of water, they would automatically form a cell membrane. Now, it would not be a cell membrane that had all the complexity that you see here. It would just be the heads would point out toward the water and the tails would point in away from the water, because the tails are hydrophobic. They're afraid of water, and the heads are hydrophilic. Remember that the heads are hydrophilic because phosphate is there and it's ionic. It has a charge, and water is attracted to anything that has a charge. It's not interested in the nonpolar tails, but the water will pull close to the hydrophilic or the phosphate heads. All right, and then I guess that's it. So then the last thing that we're going to talk about with the lipids are the steroids. Steroids include one class of our hormones, and they include cholesterol. The steroids are notable because they're a whole bunch of fused rings, so they look different than anything we've seen before. It's a whole bunch of these rings that are they share bonds. They're completely fused together. And then the presence of different functional groups is what leads to the different functions. You don't need to know a whole lot more about them right now, but you need to learn to recognize them. So they look different than the things that we've seen before. Here's cholesterol, but other ones that you might recognize would be progesterones. There's testosterone down here. The estrogens are here. I think those are mostly the ones that you would recognize. So those are the lipids. There are three types of lipids. There are the triglycerides. Remember, three fatty acids attached to a glycerol molecule. There are the phospholipids that make up our cell membranes. And then there are the steroids that are these fused rings, and they can be hormones or cholesterol in that case. All right, that is it. We're going to stop here. You can shut me off now, and then we'll start up with proteins in the next set of notes. Okay, thanks. Bye.